Hello everyone, today I'm going to be discussing light-dependent reactions in photosynthesis, specifically entailing about electron transport chain. And today we're going to discuss how electron transport chain works and what are the products out of it. So the sole purpose of electron transport chain is to make the necessary compounds for energy in order for other functions to work, such as the Calvin cycle. And the sole purpose of this energy is to push out these activators. They're called activators, to create a cycle for our plants to get food, and very necessary for sustaining their lives. Their plants, they're living things too. And similarly, as humans, we also need food and we also need energy in some sorts. So that is the basis of how the electron transport chain works, and the products of which are the NADPH, which I'm gonna be discussing later, and the ATP. And this is the two products that we need. And we start off with what is the structure of the thylakoid, like the thylakoids. What do you call this one? Yes, the thylakoid. And the thylakoid starts off with this thing over here, and that's what we call the thylakoid membrane. That's what separates the internal and the external environment of the thylakoid. And the thylakoid comprises of a lot of proteins, and this is also a bilayer. It has this thing and this thing, which I'm going to be discussing, and that's basically it. It's just to separate and to create something out of it. Okay, sorry. And the external environment is what we call the stroma. It is the aqueous liquid. Is a liquidy stuff there. There's a lot of compounds floating around there. It's just like that, and that's where Calvin cycle takes place, because it's just floating out of nowhere. Like, it's literally outside. Think about it. And it's comprises with protons, inorganic phosphates, or whatever. It's something like that. And the inside is what we call the lumen. So the lumen is basically is the internal, which comprises of hydrogen ions that are produced or separated by this thing over here, which I'm going to be discussing. And we start off with photons. Photons are very essential for activating these electrons inside of what we call the photosystem 2. Now, photosystem 2 is basically another protein of which it carries bound electrons, and these electrons, once they get hit by these photons, these things gets excited. In that way, these things gets excited. It moves to a higher energy level. And in chemistry, I think it's something like this. Maybe think about it like this. So this thing over here is gonna move there because it's much more higher energy. Yes, it's gonna move to this area here. It's something like that, similar to that. And so what happens is, once it moves to this higher area over here, it's gonna get to this thing. This photosystem 2 is gonna lose electron, and it's gonna release the electron outside the chlorophyll, and it's gonna get carried by this peripheral protein of this blue thing over here, of what we call plastoquinone. Plastoquinone is a type of electron carrier, And yeah, I want to explain deeper of on what plastoquinone is. It's just simply a electron carrier that helps to carry out electrons, obviously. And yeah, so I like to discuss. Um, I like to discuss on what is the photosystem too, and sometimes most of the people call this the, what do you call this one? The P. It's P6080. I don't know. Yeah, some people make things complicated, like, but it is a special chlorophyll that's absorbs this thing, and I'd like to clarify that the photosystem too is not protein, but rather it's a special chlorophyll, and it absorbs light. As we know, this is a pigment that absorbs light, so it absorbs these light waves. Um, these light waves absorbs red and blue, and it reflects green light. That's why we see most of the plants greeny, you know. And the reason why it's called P680 is because 680 nanometers in the wavelength is basically, you could get red light. This is the maximum light that you can get. I don't know, that's what they say, and that's it. So I don't know if that's irrelevant information. I just want to share it to you guys. So let's continue on what happens on the electron carrier. So this bad boy is gonna carry. It's gonna carry here. It's gonna go here, and it's gonna transfer this electrons, travel out here. Shroom, it's gonna go there, and it's gonna travel the electrons to this cytochrome, or what we call the cytochrome B6F complex. It's really complicated, but I just want to call it cytochrome. It's a complex protein, and basically what happens here is that once it travels, it travels through the cytochrome, such that it also pumps the. Since we know that the stroma also carries protons or hydrogen ions, it also pushes it there, and basically that's it. And it's gonna push down the electrons to lower energy, and no, actually the hydrogen ions is gonna move there because of the presence of electrons, creating this osmosis thingy. Yes, and that's basically it. And wait, hold on, let me change it, because that's, so yeah, this thing is gonna go down there. It's gonna accumulate as the more electrons that move here in the cytochrome. It's gonna be a lot of hydrogen ions here. Now, essentially, what happens when there's a lot of these hydrogen ions in the lumen? It's gonna create a gradient. What I mean by gradient is there's gonna be a higher concentration of protons in the lumen. Protons are the hydrogen ions, okay? Just to be clear. Essentially, when there's a lot of concentrated protons here, and there's gonna be a low concentration of hydrogen ions, this thing will get acidic, the lumen, and stroma is gonna get more alkaline because there's no ions that are activating in the system. And essentially, this is gonna go crazy. They're gonna go crazy because it's so tight, you know. They're gonna move crazy, and they probably might go here, which I'll talk about later. That's basically it. And oh yeah. So so we're gonna talk about this. This thing over here is essential, and this electron is gonna move here. Now it's gonna move to another electron carrier of what we call a plastocyanin. Yes, another same blue guy over here. It's gonna carry here to the photosystem 1, and yep, it's gonna carry down. It's gonna go to high to low. Okay, high and going down to low. It's like anaerobic, but like electron transport chain is electron transport chain is basically an aerobic. It requires something, and yeah, it you see that it goes down like there, like it goes down there to there. See, it's gonna go down here, and now that means it's losing energy. So once this guy over here stops moving, I mean like goes here to the lowest energy, to the photosystem 1, it's not gonna be activated. Similar to the thing here, it's gonna be here. It's not activated unless it gets hit by the photon, and photosystem 1 comes in to play. And what happens is it needs another photon or sunlight. So what's the photosystem gonna do? The photosystem 1, I mean, it needs another light again. Yeah, exactly. It's gonna get hit, and once it gets hit, it's gonna get excited again. The electrons are gonna get excited and moves to another energy level, in which they go here, and it's another, yeah, you guessed it, electron carrier. And basically, the other word for photosystem 1 is it's P700, because 700 nanometers again can produce red light. The red light is the strongest wavelength. No, not the strongest wavelength. The maximum wavelength that you can absorb. That's what they say. I don't know. And that's the same case of photosystem 2. And yeah, so it goes to this electron carrier of this is called ferredoxin. It is an iron sulfur, I think. Yes, this is an iron sulfur, and this is another electron carrier again, a peripheral protein. Essentially, it's gonna prepare to create the NADPH, but it requires an enzyme in order to synthesize or to reduce into something. So we have this guy over here. This guy is called NADP reductase, an enzyme, because there's A's. Remember, if it's an enzyme, there's gonna be A's there. And basically, since there's a lot of, since I talk about stroma being liquidy stuff and carries a lot of things, it also carries NADPH and NADP. I mean, the NADP. And since enzymes wait, they're just gonna wait there and like, oh hey NADP, come here, and like there it's gonna come. And also we have hydrogen ions, or the protons, and it's like, oh hey hydrogen ions, come here, so we can combine them together. And since we have electrons, it's gonna activate it, like increase the activation level, and then they're gonna combine into one. They're gonna get reduced, and that's gonna be in a result of, so like this, it goes right here, the electron is gonna activate the site, and the thing is that the H plus and the NADP is gonna combine into one, and that's what makes it good, because you're gonna get another hydrogen carrier, a proton carrier, which is important for the other functions, which is what we call NADPH. And I don't want to say the abbreviation for this. Hell no, it's really damn long. But NADPH is really important to carry the proton, which is the H. This guy over here. It's gonna be extracted in the Calvin cycle. Very important, by the way. I don't know. I just say very important because that's basically the simplest term I can say. And it just produces, helps them, give them, activate them, or whatever. So the sole purpose of the NADPH is, hold on, sorry, is basically carry these hydrogen ions. That's simply it. And now we're gonna set that aside for a bit, since we have the products already. There you go. Now, yeah, that's the purpose of enzyme. That's why enzymes are really good when it comes to synthesizing or decompositioning stuffs or whatever, to break down or to mix up something, and needs these things to and electrons activate the stuff. Is really good. That's why electrons are really good. Yeah. And okay, ferredoxin is gonna carry this thing, and then it's gonna activate this. So the abbreviation of NADP is what's that again? Nicotinamide adenine dinucleo. Nicotinamide adenine. Wait, hold on. Nicotinamide adenine dinucleotide phosphate. Yes. And the sole purpose is, and this is very essential. Yeah, I just said the sole purpose. Sorry. Is really good for anabolic reactions. Yes. And okay, since we're done making the NADPH, and what's gonna happen is we're gonna entail about ATP and how is ATP produced. So yeah, let me set aside this high concentration, whatever. There you go. Let me put it there. Yeah. So here, and as we all know that there's one issue when it comes to moving the electrons to this stuff here. Let's see. Since the electrons are gone, like here. It was earlier here, right? Let me get this guy over here. Then this guy is like, oh. Now the electron is gone here, and in the photosystem too, when there's no bound electrons inside it, essentially what happens is it's gonna go unstable, or it's gonna destabilize itself. That's why it's gonna call out the water molecule, like, help, I need something, help, help. So essentially he's gonna call out the water molecule, which is this guy over here. Yes. Wait, hold on, let me go down. And now what essentially he's gonna do is like, what should I do with this water molecule? I'm gonna get the electrons out of it. So he's gonna split the water molecules, and what happens is it's gonna spit out 1/2 of the oxygen. And the reason why it's 1/2 is since this is one singular oxygen atom, it's just 1/2. Yeah. But I don't like seeing 1/2 in chemical equations. I just don't like it. So okay, let's back to our topic. So yeah, the hydrogen, there's gonna be 2 hydrogen ions, and okay, there's gonna be 2 electrons. Okay, this is the product that we want in order to stabilize the photosystem too. Okay, now since I don't like seeing 1/2 here, I'm gonna remove that, because we're gonna add 2 molecules of water, and essentially what's gonna happen is it's gonna make things relevant again, and I'm very happy. There you go. Yeah, there's gonna be 1 oxygen atom, and there's nope, there's not 2 but 4, because we have 2 now, 2 hydrogen ions, and it's gonna be multiplied here. So you have 4, and we're not forgetting about electrons. We're gonna have 4 too. Okay, it's gonna be oxidized into 4. So that's the equation. It's basically oxidation now. Okay, let me put that there. Keep in mind, guys. Oh, that's really small. Now you have a product of the oxygen, right? This oxygen is gonna go out Obviously, this guy needs to go out. And right. Okay, so now we have this. And now these four things over here, since we produce this thing, let's say we have this. Oh, that's pretty bad. There you go. And as we all know, we just pump a lot of hydrogen ions here. It's because of the electrons moving here, and then it's such that it just pushes the protons inside the lumen. So yeah, that's what I said. It's gonna create a gradient. There's gonna be acidic air here, and it's more alkaline. Now, what happens when it's acidic? It's gonna shake. It's gonna find another way to escape because it's very high concentrated here. So it's like, I need to get away. Where should I go? Where should I go? So they go to this another enzyme, a channel, and these hydrogen ions are gonna travel right there. And this is what we call the ATP synthase. It is an enzyme. Yes, it is an enzyme. Of course, it's an enzyme. And essentially, it's something like the great example that I can give you is it's like a generator, you know, when you create something like, you know, example, if I do mechanical energy, I mean mechanical stuffs like pushing or pulling, and it's gonna spin the motor out and then create energy or whatever. Yeah, it's basically like that. So once the hydrogen ions pushes through the ATP synthase, it's gonna technically spin out the enzyme is gonna rotor out such that the outside or the stroma is gonna mix the ADP around it. So okay, I haven't told you guys. There's ADP around the stroma. It's literally floating there. It's a liquid, same as the NADP plus. It's floating around there. It's because of the Calvin cycle. Okay, it's releasing those stuff. I'm gonna be discussing that later on why it's releasing stuff in the stroma. And yeah, so this now we have the ADP. No, it's not ADP plus the inorganic phosphates. Now, what happens when we combine them together? Well, obviously, we're gonna make ATP. And yeah, this one now we have this, and simply, you know, this hydrogen ions gonna go here back, so it's just conserved. It's not gonna go away, like it's gonna go boom, it's gonna be destroyed. Conservation of energy. So yeah, these guys can't like just conserve. It's not gone. Like once this hydrogen ion is gonna pass this synthase, it's not gonna be magically gone, but rather it's just gonna move there and it's just gonna spin the synthase and then like there, and now they're traveling, and they might get stuck in this cytochrome. Yeah, so go back in a cycle over and over again. It's just a cycle, and most of these stuffs are not gone except the oxygen, which will be released to the atmosphere. And yeah, so okay, let's go back to how ATP is made. I'm going out of way. So yeah, the ATP, you already know what's gonna happen, right? If we combine them together, well, obviously we're gonna make ATP. ATP is very essential. Yep, ATP. Now, ATP is basically the energy currency of the system. Very important when it comes to making products such as glucose. Is to activate the Calvin cycle into creating these phosphate groups or whatever. I'll be discussing that too, because that thing's really complicated too, but I'll try my best. So this is gonna be the product. Yeah. Oh. Bad. Alright, so here you go. Okay, that's simply it. That's how it works. I'm gonna summarize this for a bit. So in order to make NAD pH, it's gonna get hit by this light over here. Yes, dang. It's gonna get hit there, and the electron is gonna go up, and it's gonna slide down to this photocannon, the plastocannon. It's gonna go down there. It's gonna travel to this cytochrome, and it's gonna travel. And once it travels here, it just it pushes the protons from here to create something. It's gonna be a lot of protons here. It's gonna be less protons here, and the electron's gonna still gonna travel to the lower energy level, to this the plastocyanin, the carrier again. It's gonna move here, and since there's no energy now, it's gonna get hit by the sunlight again. It's gonna get hit there, and it's gonna go up again. It's gonna go to this another electron carrier as the ferredoxin, and it's gonna go to this enzyme, and it's gonna combine with the external compounds of a NADP plus and the protons, gonna make NADPH. And for the ATP, it's gonna undergo to a process of photolysis, and essentially it's just gonna split water because there's no electrons here, so it's gonna take the electrons inside the water there. It's gonna be there, and the cycle goes over and over again. Like there, if there's no electron here, it's gonna be gone out, and yeah, it goes here. The other hydrogen ions is gonna go to this lumen. It's gonna be high concentration. If there's high concentration compared to the other stroma, this guy's gonna move here because that's the only way out, and it's gonna spin out this thing. This is what we call chemiosmosis. Chemiosmosis is basically the process of the ATP synthase to create ATP. It's just to spin out things. This is also osmotic way passage, because it just goes there to high to this movement here. Yeah, that's basically it. It's wait, I think this is a facilitated osmosis, because this is high to low. This is osmotic push, you know. It's a facilitated, yes. Yeah. So that's basically what happens. And alright, let me label this up. This is chemiosmosis, alright? Chemiosmosis. Let me type that around there. Chemiosmosis, and that's photolysis. There you go. That's basically it. Yeah, I can sum it up for that. And like, this is a cycle, like I said. Once the hydrogen ion's gonna go here, it's gonna stay to the stroma again. It's gonna float there. And now, if there's electrons passing there, it's gonna pump out the protons, move here to the lumen again. The cycle goes over and over and over and over again. And the water, yes, you technically need to water your plants, guys. You need to water your plants, so it does this stuff here. So it's gonna give them electrons, you know. It's gonna activate the chain. That's why water is necessary. And oxygen is also necessary, of course. As humans, we need them. And yeah, and also, some people may ask, where does this inorganic phosphate come from? Technically, it comes from the nutrients in the soil. It's basically the same as water. It's gonna get absorbed same as water. So the water goes from the roots and it's gonna be distributed to the plants, leaves or whatever. And same thing as the inorganic phosphate. That's why fertilizer is a must when it comes to planting stuffs. So yeah, that's just a basic knowledge on why there's inorganic phosphate, because of the nutrients of the soil, and it's gonna get absorbed by the roots. It's gonna go up, and oh, I can make use of this phosphate. That's why different minerals is very important in plants because they can make use of something. So yeah, that's basically it. I'm gonna be discussing about Calvin cycle later. Thank you guys for watching. I hope you like this, and I hope you understand this because I'm really bad at teaching. And thank you guys.